Let us see the function of the second domain of DNA polymerase, and the second domain is the fingers domains. So the fingers of the polymerase are also important for catalysis of the reaction. Several residues located within the fingers bind to the incoming DNTP. So in these fingers, several amino acid residues are present. These are important amino acid residues, and they bind to the incoming DNTP. So they have role in the catalysis. More importantly, once a correct base pair is formed between the incoming DNTP and the template, the finger domain moves to enclose the DNTP. So initially this is open, but when a correct DNTP comes and it makes the base pair with the existing template nucleotide, after this base pairing, this finger domain moves and it encloses this DNTP. This closed form of polymerase hand stimulates catalysis. Then the nucleophilic attack starts and catalysis proceeds. In contrast to the fingers and the palm, the third domain, which is thumb, is not intimately involved in catalysis. So although it has role in the catalysis, but directly it is not involved in the catalytic process. Instead, it interacts with the DNA that has been most recently synthesized. So the most recently synthesized DNA that interacts with this thumb domain. This serves two purposes. This thumb domain this interaction serves two purposes. First, it maintains the correct position of the primer and the active site. You know, this is the active site. This palm is the active site of DNA polymerase, and so it maintains the correct position of primer and the active site during catalysis. Secondly, the thumb helps to maintain a strong association between the DNA polymerase and its substrates, that is DNA. So it is also responsible, this thumb is also responsible for the strong association between DNA molecule and DNA polymerase. Due to this association, this association contributes to the ability of DNA polymerase to continuously add many new DNTPs to the existing template.